Okay, so here, gentlemen. Yesterday, when I introduced the course and started on talking about the barriers that make up our body that protect us from outside threats, the outside environmental pathogens. So in today's lecture, I'll talk about the internal cells that make up this innate immune system. And so the objectives of this lecture is to talk about the different cells that we classify as part of innate immunity, to talk about the functions of each group of cells, to describe how they recognize what we know as self versus non-self, and to talk about how they function, how they perform their different actions, and then finally to talk about this unique set of innate immune cells, which is like a hybrid between adaptive and innate immunity, in that they're important in protecting us against virus infections as well as cancer. And these are called natural killer cells. So just to repeat from what I said yesterday, we defined the term innate as inbuilt. So it's not able to adapt. It's what we're born with. It's a first responder. So as soon as you get infected, these things come into action, and they have relatively broad specificity, as opposed to the adaptive immune response, which is able to evolve to provide stronger protection. Because of that, the adaptive response takes longer to kick in, a couple of days, compared to hours with regard to the innate immune system. And so the upshot of this is that the innate immune system acts as a glue to trap the first infection into the body and try to remove as many of the microorganisms as it can, and then allow for the adaptive immune system to come and clear up. But these two functions, the adaptive and the innate, aren't completely separate. They're very linked in that activation of the innate immune cells through the production of these cellular messengers, which we call cytokines and chemokines, acts to activate the adaptive immune system and draw those cells to the site of infection. So they're very closely related, even though we talk about them as separate entities. Another thing we need to bear in mind is that the innate immune system, as well as defending us against invasion from microorganisms, also plays an important role in the day-to-day maintenance of health in our bodies during development, for removing cells as they go through these genetic programs of growth, proliferation, and then apoptosis, or programmed cell death. So with regard to the cellular components of innate immunity, I don't know if you did hematopoiesis last year. So just a revision, that within our bone marrow, we have these so-called stem cells, which are the progenitors of all the different blood cells in the body. From these stem cells, as they differentiate, they become more committed to particular lineages. And so those within the myeloid lineage will differentiate into these three classes: neutrophils, eosinophils, and basophils. These three are called together, are grouped as and called granulocytes. They're called granulocytes, obviously, because if you look under the microscope, their cytoplasm is filled with these dark granules. Related to these granulocytes is the myelomonocytic lineage, which make up the macrophages and the monocytes. The term monocyte and the term macrophage refers to the same thing. Only thing is that the monocytes are more immature and they are what circulate through the blood. Once they see antigen and get activated, they will then differentiate into what we call macrophages. Then within the cells of the adaptive immune system, we have the B lymphocytes, which are responsible for making antibodies, and the T lymphocytes, which are divided into two groups, what we call the T helper cells and the cytotoxic T cells. And as I said, the natural killer cells are like hybrids between the innate and the adaptive. So they have characteristics which are similar to the lymphocytes, but their recognition system is more non-specific, and so we classify them as innate cells. So this is just repeating what I just said, that within these granulocytes, we have neutrophils, eosinophils, and basophils. The monocyte macrophage lineage, the macrophages, there's a class of them that when they get differentiated, instead of circulating through the blood, they are fixed tissue macrophages. And there they're given different names. So within the liver, you have macrophages there that are called Kupffer cells. Within the brain, you have a lineage of macrophages which are important for the maintenance of health in the brain, and these are the microglial cells. And then the lungs, we have alveolar macrophages. Within the spleen, the splenic macrophages, and in the gut, peritoneal macrophages. Then we have another important class of cells which are like the key in bridging the innate and the adaptive. And these are the dendritic cells. They perform similar functions as the monocyte macrophages. And so for a long time, there was a debate whether these two weren't the same lineage of cells, but now they're regarded as a separate lineage. And then we have the natural killer cells. So in addition to morphology and lineage, we can also classify these innate immune cells based on their function. So within those cells that can eat foreign cells, like bacterial cells, which we call phagocytosis, there are three groups of cells which can do this, and these are the neutrophils, the macrophages, and the dendritic cells. The cells which are can directly kill virus-infected cells or cancer cells, these are the natural killer cells. And then, as I said, these innate immune cells, which are responsible for activating and drawing in the adaptive immune response, these are first and foremost the dendritic cells, and then the macrophages, and also the natural killer cells, because they are also a major source of these intercellular messengers, these cytokines. So the major source of cytokine reduction. And then we have four cells, three of which are granulocytes, namely the eosinophils and the basophils. And these we won't deal with much in this early stage. We'll talk about them more in hypersensitivity because they're more associated with allergic responses. Mast cells are important because they are one of the key activators of the inflammatory response, which you'll hear a lot about from your pathology lectures. So this is a cartoon, well, a video of a neutrophil chasing some bacteria. And then phagocytosing. So here, what has happened is that that neutrophil has flowed its cytoplasm around that bacterial cell and then enveloped it to take it inside. You can divide that video into different processes. So the first is that with that neutrophil chasing that bacteria, there has to be some kind of recognition. Why is it chasing that bacteria? It's because that bacteria is sending out signals, I'm foreign, I'm non-cell. So there has to be some kind of ligand receptor activation going on there, right?
Are they the cytokine?
No, these no, because then what you're talking about is that the bacteria would be releasing cytokines. Bacteria don't release cytokines. They release chemicals, right? So we break down those processes. So the first is that the bacteria or the phagocyte. So when I say phagocyte, I'm talking about a cell that is able to perform phagocytosis. So as I said, there are three kinds of cells that can perform phagocytosis. So it's the neutrophil, the dendritic cell, and the macrophage, right? So the first instance is that this phagocytic cell needs to bind to that target bacterial cell. So you need some receptors on this phagocytic cell sticking to the surface of that bacterial cell. Once it's done that, it flows its cytoplasm around that bacterial cell and envelops it and brings it into the cytoplasm in this vacuole. Within the cytoplasm of this phagocytic cell, you have pre-existing vacuoles, which are called lysosomes, which contain various proteolytic enzymes. So as this bacterial cell enveloped within this, what we call a phagosome, shifts through the cytoplasm, the phagosome will fuse with one of these lysosomes. And in this fusion, that will release the contents of the lysosome into the phagosome. So we have what we call a phagolysosome. Within this phagolysosome, then, through the action of these enzymes, they begin degradation of that bacterial cell. So they break down that bacterial cell into small components, small pieces. Some of those pieces will then get excreted from the cell, but then other components will then be loaded onto particular structures on the phagocyte and then expressed on the surface of the phagocytic cell. This I will talk about in more detail in a later lecture, because this is the key process whereby the adaptive immune system gets activated. This process is called antigen presentation, because in this process, these various components of that bacterial cell are presented to the T T helper cells, to the T lymphocytes. So first of all, that recognition step, what are the structures on the bacterial cell that are seen by these receptors on the phagocytic cell? These are what we call pattern-associated molecular patterns, or PAMPs. What do we mean by patterns? So first of all, tell me, I'm sure a lot of you will know, what is the structure, the biochemical structure that is recognized by antibodies? Sorry? Yeah, the antigens, but what is the antigen made up of? Proteins, right. So what are the three basic macromolecules? Carbohydrates, lipids, and proteins. And what do you know about carbohydrates and lipids as opposed to proteins? In terms of their fine structure, what are they composed of? They're composed of identical molecules that make up these repeated chains, these long chain fatty acids or long chain complex sugars, sugar molecules, right? So as opposed to a protein, which is made up of amino acids, and depending on the composition of that amino acid will depend on how the protein folds. So the proteins look very different in terms of structure. They might look like a sheet or a rock or whatever, right? But the carbohydrates and the lipids just form these long repeated chains. And this is what we mean by patterns. So on a bacterial cell, where would you expect to find most lipid, or on any cell? On the cell wall and the membrane, right? And the other thing too is that because these never vary, because if they did, the bacterial cell wouldn't be viable. It wouldn't form a viable membrane. So these are set structures on a bacterial cell. So these are the structures that these receptors recognize, which we call pattern-associated molecular patterns, or PAMPs. Within these PAMPs, we divide them into two groups. The one which was associated with bacterial cell walls, or fungi, or yeast, or viruses. And we call these microbial-associated molecular patterns, or MAMs. But now here's another new bit of thinking you've got to realize. If you get sunburn, for instance, and I'm sure you've all experienced sunburn, your sunburned skin feels pain, it feels redness. Yeah? Yes. All of that is basically an inflammatory response, the same inflammatory response that you get when you get a bacterial infection, or you puncture yourself with a contaminated nail or something like that. The same processes are happening in your body. What is happening within the UV exposure scenario is that the high energy UV radiation kills the healthy cells in your body, and those dead cells burst or lyse and release intracellular contents. Those intracellular contents, which are DNA, RNA, mitochondria, get recognized by nearby cells as there is cell damage going on. And so they initiate the same inflammatory response as what you'd get if you got infected with a bacteria. And we call that sterile inflammation, because it's the same inflammatory processes going on there. So this is why we call these damage-associated molecular patterns. So this kind of cell damage happens without any microbial invasion. This can happen in case of trauma, if you hit your arm too hard and so you burst some cells. Whenever intracellular contents of cells are released, that is picked up as danger signals. So this is a diagram of the structure of cell wall of the bacteria. So here you have the membrane, cytoplasmic membrane, and then these repeated chains of sugars and lipids. In this case, this would be a gram-positive bacteria. And here you'll learn more about this in your microbiology lectures. These are lipoproteins on gram-negative bacteria, and these are the teichoic acids on the gram-positive. These repeated units of structure. And these are seen by the pattern recognition receptors on phagocytic cells. Now, but another thing we've got to understand, well, as I said, this is the same as what I've talked about in terms of the DAMPs, or the damage-associated molecular patterns. So the intracellular contents of cells, when they get lyzed and release nucleic acids like DNA and RNA, mitochondria, if these get picked up by PRRs of nearby cells, they will induce those cells to undergo pro-inflammatory responses. So these pattern recognition receptors, there's several, but the three major groups are known as toll-like receptors, nod-like receptors, and rig-like receptors. These receptors can be expressed either on the outside surface of the phagocytic cell, but also internally within these cells themselves, within the vacuoles, or even within the cytoplasm. The consequence of that is that you need to then realize that cells themselves, within the cell, there is an immune system going on. That within every cell, whether it's a macrophage dendritic cell or an ordinary fibroblast, there is an internal immune system which is able to detect that any, for instance, a virus getting in doesn't belong there. That is non-self. So cells themselves have an internal immune system. And this immune system is activated through the recognition or engagement of these PRRs. So with regard to the external PRRs, one of the first PRRs that was detected was one of these toll-like receptors called TLR4, which is on the surface of a macrophage. TLR4 recognizes lipopolysaccharide. What do you know about lipopolysaccharide? Where is it expressed? Bacteria. What kind of bacteria? Gram-negative bacteria, particularly E. coli. E. coli, when it goes to, if you get infected with gram-negative bacteria like E. coli, and it gets into the bloodstream, it can cause endotoxic shock. That endotoxic shock is due to the fact that these circulating lipopolysaccharides on the surface of these gram-negative bacteria get picked up by the macrophages and stimulate the macrophages to produce these inflammatory cytokines that circulate through the blood. These inflammatory cytokines are so potent in causing cell damage, blood coagulation, etc., that ultimately a person can die from multiple organ failure because there's been so much activation and production of these inflammatory cytokines as a result of the systemic exposure to this lipopolysaccharide. So that's one example of how potent this whole system is in terms of activating these innate systems. Within the toll-like receptors can be expressed or mostly expressed on the outside of cells, but there are also other toll-like receptors internally, like in the vacuoles within the endosome of the phagocytic cell. The other key or the other major PRRs are the nod-like receptors, which we call NLRs, and the rig-like receptor. Now, the rig-like receptor, of which there are 3 or 4 different kinds, they can recognize viral RNA like coronavirus. A coronavirus is a single-stranded RNA virus. When it gets into a cell, all single-stranded RNA viruses, when they get into infect cells, they need to make a double-stranded RNA intermediate in order to make new baby viruses. Eukaryotic cells don't have double-stranded RNA. So as soon as double-stranded RNA appears in the cytoplasm, these receptors can pick that up and say, that doesn't belong here, that is non-cell. And that will activate that cell to make a potent antiviral cytokine, which you'll learn later, called interferons. Another key feature of one of these other receptors, nod-like receptor, or NLR, is that if it picks up a foreign MAMP or PAMP, it causes that nod-like receptor to aggregate into this structure called an inflammasome. This inflammasome is a multi-proteolytic enzyme complex, right? So once this structure is formed, now consider that there are many enzymes that are preformed but inactive. They are inactive because they have, think of like a spring that gets closed up. So you have a non-functional enclosure of this enzyme, right? What this multi-enzyme structure, the inflammasome, does then is that it takes these inactive enzymes and cuts off this regulatory region. Once that's cut off, that enzyme can spring open to become a functional enzyme. So within these macrophages, there are 2 of these preformed inactive enzymes. One is called interleukin-1 or IL-1. The other is called interleukin-18 or IL-18. So in a normal healthy macrophage, you have these pre-made IL-1, IL-18 cytokines, but they're inactive because they're enclosed by this regulatory portion of the protein. When you get an invasion and a threat, and so engagement of these nod-like receptors which then aggregate to form the inflammasome. These inflammasomes cut that inactive portion of IL-1 and IL-18. That then becomes a inflammatory cytokine. You get then active IL-1 and IL-18 being released. And once they're released, they can then start the chain of inflammation, the inflammatory response. So this is how inflammation begins as soon as you get invasion by these foreign particles, by these foreign cells. So with regard to the types of cells which can perform phagocytosis, the major one is the neutrophil. We know that because the neutrophil makes up about 60% of the white blood cells in your blood. And so this is the first responder. As soon as you get invasion by bacteria, these neutrophils will come and phagocytose or eat those bacterial cells. Recall that diagram I showed about phagocytosis. Those phagocytosed bacteria get in, fuse with the lysosomes within the neutrophil. The lysosomes release the enzymes and then break down the bacteria. But when those lysosomes are released, just like a bee that's already stung somebody, when they are released, the neutrophil itself gets killed. The neutrophil dies as well and releases its contents to the outside. These are called nuclear extracellular traps because the intracellular contents of the neutrophil, a lot of that contains DNA, which is sticky. So that acts like a glue in being able to trap a lot of other bacteria within that site. But not only that, these intracellular contents of the neutrophil then act as damps or damage-associated molecular patterns, because then the DNA and RNA of the neutrophils can then be picked up by PRRs by surrounding cells and activate them to undergo an inflammatory response. So in this way, the neutrophil then can be thought of as a first responder in trapping extracellular invasions, but then also acting as a sensor system to activate surrounding cells to amplify this inflammatory response. The monocyte macrophages are also phagocytic, but in addition to phagocytosis, they have an important function in that they are a major source of cytokines, these inflammatory cytokines. And also within these monocyte macrophages is where you have these inflammasomes being formed, which can release these active pro-inflammatory cytokines like interleukin-1 and interleukin-18. All of these immune cells express unique surface proteins. And we can then identify what type of immune cells are present within the body or within an inflammatory site based on the expression of these proteins. So in the case of the monocyte macrophage, they are characterized by the expression of CD14, surface protein CD14. I'll explain to you in a later lecture why we call them CD. So you have CD3, CD4, CD8, and so on. So with macrophages, CD14 is actually one of the accessory proteins associated with that PRR I told you about, TLR4, which recognizes LPS on gram-negative bacteria. So the as well as phagocytosis, production of cytokines, the other major function of the macrophage is this process of antigen presentation, cutting up that bacterial cell into small peptides, which can then be loaded, presented on the surface to be seen by cells of the adaptive immune response, particularly T lymphocytes. And then another function of these macrophages is that once the inflammatory response has dealt with the threat, the invasion, you have another class of macrophages, again through the production of particular cytokines, which promote repair of damaged tissue, to promote wound healing and repair, and also to block any further inflammation to go on. Dendritic cells are as I said, have similar functions to the monocyte macrophages, but they are specialized antigen presenting cells. They are totally dedicated to phagocytosing foreign material, chopping them up into individual peptides, and then presenting them to be seen by T lymphocytes. They do this because they have these long processes. They're called dendritic cells because they look like dendrites in the neural tissue. And because they have long surface, it means that they can express lots and lots of receptors, lots and lots of antigen. So within this dendritic cell, you can see lymphocytes, multiple lymphocytes bound on there on the surface, picking up antigen that is being presented by this dendritic cell. They are the key activators of the adaptive immune response. So finally, I'll talk about these natural killer cells. And this is a electron micrograph of a natural killer cell, which you can see is quite small in relation to a tumor cell, where the natural killer cell has attached to the tumor cell. And this basically shows here that you have a tumor cell, and these natural killer cells stick to it. And what they do is that they inject granules into the tumor cell, causing that tumor cell to undergo programmed cell death or apoptosis. But unlike the neutrophil, they do not die and they can go on to kill other targets. Yeah, you've seen that one. So the question, so that is their main function as natural killers. We know that they were first discovered because there are some kids who are born with the inability to produce natural killer cells. So they don't have natural killer cells in their immune system. And what we find from these kids is that they're susceptible to a lot of virus infections. So that tells us the importance of these NK cells in combating virus. How do they recognize that a cell is infected with a virus compared to a normal cell? This comes with regard to what's called the missing self hypothesis. And here I will introduce this concept of MHC or HLA protein. Have you ever heard of? So what are HLA proteins? Right. So HLA proteins you can think of as barcodes in our bodies that tell our immune system what is self and what is non-self, right? Based on HLA, it will determine whether we can accept a liver or a kidney graft from someone if it's a close relative. If we are genetically identical, the HLA is compatible, our immune system will accept that graft. So basically, the natural killer cell in a healthy body sees normal, healthy HLA proteins on cells, and that provides a negative signal to the natural killer cell to leave those cells alone. You get some viruses that if they infect a cell, they can block the expression of HLA on the infected cell. They do this because they want to hide from these cytotoxic T lymphocytes, which can see virus proteins on HLA and kill them. So basically what they do is they try to hide from the cytotoxic T cell response. The natural killer cell acts as a backup system because when it comes and sees that this cell is not expressing HLA, it then kills it. And this is why it's called a missing self hypothesis. Similarly, if you have a mutated cell, so this is not infected with any microorganism, but it has it is developing into a tumor cell, and through that mutation, it's causing misfolded or mutated HLA molecules. The natural killer cell can see that mutated HLA, recognize that as not being normal, and kill it as well. And that's why natural killer cells have a major role to play in this process that we call tumor surveillance. So finally, this is just a summary of the composition of white blood cells in the blood with regard to proportions. So neutrophils, as you can see, make up the biggest number of cells in the blood, and that reflects their importance as first-line responders in any form of invasion. The next are the monocyte macrophages and the natural killer cells. And then we have these smaller numbers in terms of eosinophils and basophils, reflecting their role in allergic responses. Okay, that's it. Are there any questions?
Regarding the numbers, can we memorize an approximation?
No, you don't have to know the numbers. This is just to give you an idea. All you have to know is that neutrophils make up the largest proportion. Right?
Then lymphocytes, right?
Sorry?
Lymphocytes are not here, but then
No, because I'm just dealing with the innate immune cells. Lymphocytes make up about 20%. What is the difference between the phagosome and the phagolysosome? So the difference is that the phagosome is at the earliest stage. The phagosome is at the stage where the phagocytosed bacteria has been brought into the cytoplasm, but it hasn't yet fused with the lysosome. Within the phagolysosome is where you get breakdown of that phagocytosed bacteria. Any other questions? Okay, that's it. You want to take another attendance? Hang on. Sorry? Oh, okay, sure.